I told you in my initial words what this building is and what the very general ideas about what we're doing, but I haven't told you what we're actually doing. And this is what we want to do in the next 25 minutes or so. The 5 main PIs of this building will present their research in a very short and concise way, and we do this in alphabetical order. That means we start with Kristian Franze.
Yeah, thanks, Jochen. So, what you see here on the right-hand side in small is a human brain, or a picture of a human brain. On the left-hand side, the colorful picture shows you long-distance connections in the brain, where lots of neurons connect to places that are far away. But the brain doesn't start off like this. Initially, it's just a flat layer of spherical cells. And during development, the neurons are born and they extend these processes. They connect to far distant targets. And a question in the field has been for decades or for more than a century now, how does this connectivity arise? How does this work? And what you see here is an anatomy of a neuron. So it has a cell body which contains a nucleus with the DNA, so the genetic material. Then there is a number of short branch processes which collect information from the environment, which is called dendrites. And then there's one usually long dominating cell process, which is called the axon, which sends information to the next cell. And during these axons, actually, they can get pretty big. So axon in the neck of this guy here can grow up to 4 meters in length, so a single cell. So the question is, how do these cells know during development where to grow and where to stop growing, where to connect? So this has been puzzling the field for ages. And of course, modern biology, medicine is all focused on molecular research. And so researchers found that the growth cone, so the leading tip of the growing axon here, has a lot of so-called receptors, so proteins that can detect certain signaling molecules. And so it's this kind of a nose sniffing around for attractive or repulsive signals. So an example, when I wake up in the morning and my wife already made some coffee, then there is this lovely smell of coffee in the air. It's a strongly attractive chemical gradient to me. Right? So it's the same for neurons. And so textbooks tell you that if a neuron then encounters a gradient of an attractive cue, it will turn towards it. And this is how they are guided through the developing brain to the proper place. But unfortunately, this is beautiful, the idea is beautiful, but it doesn't always work. There's lots of examples where this model fails, where it cannot explain the patterns that we actually see in vivo, which suggests that we are missing something. So what could this be? Point is, when the axons grow through the tissue, they move. And motion is naturally driven by forces, not by molecules, right? You need a force in order to move. So the growth cone has to pull on its environment, similar to me, right? This is lovely to have the smell of coffee in the air, but I won't get anywhere near the coffee unless I get out of bed and I move my legs, right? I need to mechanically interact with my environment, and the same is true for these growing neurons. And that's what my lab is interested in. So we want to figure out how these mechanical interactions impact the development and function of the brain. In order to address this question, we exploit lots of tools from the engineering and physical sciences to measure the forces that the cells exert on their environment, to measure the mechanical properties of cells and the tissue, and also to study interactions between growing neurons and their mechanical environment. And model systems we exploit include the African clawed frog, Xenopus levis, and the fruit fly, Drosophila, which are very powerful developmental organs in model systems in order to study fundamental questions in biology. So, and using these approaches and model systems, we discovered, amongst others, that the brain is not mechanically, it's super soft, it's like soft as cream cheese, but it's not flat, it's not the same everywhere. So there is certain stiffness patterns in the brain, and it turns out that these stiffness patterns are indeed important for proper wiring of the brain. So these patterns come, and if they are perturbed, then the wiring goes wrong. And you know, wrong wiring may lead to lots of like neurodevelopmental disorders such as schizophrenia, autism spectrum diseases, etc. So mechanics is important. So this is one thing we study, and we also want to understand how does a neuron know what the stiffness of the environment is? How does this work? What's the molecules that convey this message? And then ultimately, how are these messages integrated with the chemistry that is around that is equally important? So that's what my lab is working on. And so we mostly focus on developmental problems, so how does the brain get in shape, because we need to understand basic mechanisms first before we understand what goes wrong when it goes wrong, right? But every now and then, as Jochen said in the intro, well, of course, we have an eye on real-world problems, and one of them I just want to mention here is spinal cord injuries. So after spinal cord injuries, in mammals, neurons cannot regenerate, which means that people with spinal cord injuries are often wheelchair-bound for the rest of their lives. And despite decades of research on the molecules involved in this process, we haven't really advanced in our understanding on treatment possibilities. So something is missing here again. And so we started looking into this a couple of years ago, and we found to our surprise that, so this is a stiffness map of healthy spinal cord, where red is stiff, blue is soft, and after injury, scar tissue forms. And we usually think of scars as some stiff tissues, right? In our skin, liver, some people might have heard of. So in the central nervous system, in the brain and spinal cord, the tissue gets softer after injury. The scars are soft. And from our basic research, we know that neurons grow better in stiffer environments. So it's very tempting to speculate whether, if we make the tissue stiffer, may this facilitate regeneration, or if we make the neurons blind, if we understand how they transduce this stiffness, if we make them blind, if we can pharmacologically interfere with this, may this help? And there is first exciting data that we have now where we find that indeed when we stiffen the tissue after injury in rats, then they regain function. So this is really promising and exciting, and it's even more exciting to be here, to be in this building at the medical campus at FAU. So I'm really exciting. I'm very much looking forward to working here in the future. And of course, I haven't done any of these experiments. This is a great team effort by an amazing team, full of people with different backgrounds from biology, medicine, physics, engineering, and I think that's what's needed to advance medicine. Thank you.
My research group is called Cell Physics because we're interested in the physical properties of cells, what these properties mean for biological function, and we want to find out how we might exploit them for diagnostic or therapeutic purposes. Now, I want to take you, to motivate the research, I want to take you into a supermarket. Let's say we want to buy tomatoes. It's easy enough to spot the tomatoes because they are round and red and they have this green thing on top, right? And then you stand in front of the rack full of tomatoes. What do? Exactly. Thank you very much. You do a mechanical measurement. You grab them, you squeeze them, you see how much they deform. That's a mechanical measurement, right? Because in a mechanical measurement, you might gain more information that's not obvious from the outside. The same approach is taken by clinicians, right? That's called manual palpation. They might use the stiffness or changing stiffness of organs or parts in a tissue to identify disease. Okay? We want to take the very same idea to identify disease and changes down to the single cell level. And I really like the statement by Sid Brenner, another Nobel Prize laureate, who said that developments in science depend on new techniques, new discoveries, and new ideas, probably in that order. Because a lot of the techniques that we would need to do this kind of research do not exist yet. How do you measure the stiffness of a single tiny cell? Right? So we are developing a lot of the tools that we need for our research ourselves. And out of the several that we have developed and are developing, I want to pinpoint one that is called deformability cytometry. It's actually relatively simple despite the name. The idea is if you have cells in suspension in a syringe, you use a syringe pump to push the cells through a microfluidic chip, and the chip has a narrow channel. The channel is slightly larger than the cell. Cells that we're interested in, 15 microns, channel 20 microns, much smaller than your hair, but they move at about 30 centimeters per second. And because of this, there are considerable hydrodynamic pressures and stresses acting on the cell as they passage through such a channel. So you see an initial spherical cell is first elongated, and then eventually reaches this bullet-like shape. And here at the end, we actually take then images of these cells. This is done on an advanced microscope with very fast camera. We have algorithms to identify whether there's a cell present, to detect the contour. From this, we get the size of the cell and the amount of deformation, but also a lot of other information that is contained in the image, like internal granularity, surface roughness, and a couple of other things. And if you look at the time scale, we're talking milliseconds here, which means if you let this run, we actually can measure up to 3,000 cells per second, right? And just to illustrate what that means in terms of speed, with this throughput, we can now take a drop of blood, just as it's taken from in a normal investigation by a clinician, for example. Dilute that, send that through the chip. You see the cells coming in here from the left. There's some sheath liquid coming in here as well, and we do the measurement here at the end. And if I let this run, this is still slowed down 10 times, so imagine how fast we can now assess many different cells that you find in the blood. So what we see is images like that. Again, that's a snapshot of the images we take in 0.1 second. We measure typically for 10 minutes. We have 2 million images of cells that we then analyze per patient. And there's a lot of information contained within these images, deformation and otherwise. We do many different things with that. And again, I only want to point at one particular application that came up during Corona, because we got access through our clinical partners here to patient samples from COVID-19 acute patients. If you look at the images of healthy cells, red blood cells look like this, neutrophils, lymphocytes, we can identify them. In COVID-19 patients, they looked very different. In particular, red blood cells were too stiff, neutrophils too large, and also the lymphocytes too large. And it's not too hard to imagine that if you have something that is too large and too stiff, they will not fit through small microcapillaries. And that could be at the cause of some of the pathologies associated with COVID-19 that you get thrombotic events, organ failure, and ultimate death. So to test this idea, we teamed up with Bettina Hoberger in the head clinics, Augenklinik, right next door, right there, 2 minutes. She's using OCT angiography to look at the retina and the blood flow in the retina at the back of the eye. Healthy retina, you get a lot of blood flow. COVID-19, you see the areas that are not very well perfused. And that could be due to the fact the cells are too large and too stiff. A working hypothesis that we work on at the moment. We also speculated that maybe this has something to do with long COVID, because in some patients, these changes persisted for months after the acute phase. Again, a hypothesis. But of course, that was picked up very quickly by national media. And it also gained the attention of politicians who helped support the further research. But I want to use this image also to point out the people in the back here that illustrate how closely we are already tied into the research that is going on into the local research landscape. We also used this to start a company because we just needed so many more small devices so we can do our research. And again, this is only one of the applications with one of the methods that we've developed. There's many more that we're doing. That's the team. Thank you very much for your attention.
So, my name is Benoit Ladoux. I'm French. As you can see with my accent. And my group is entitled Tissue Mechanobiology, because we are interested in how cells can move together, coordinate, and perform functions. So to give you one example that is taken from a fly embryo in vivo, what you can see here is that you have multiple cells that can coordinate, get some movement, they can migrate. There might be some different kind of movement. They can also stop moving and so on and so forth. And so we try to understand, using in vitro experiments, how we can basically mimic those kind of events. And maybe something that is that can characterize well what we are doing is the So that's the idea is clearly to have some coupling between what could happen in physical system. For instance, where in some of them you have a passive behavior. For instance, you may have collective movement in that case, which is driven by the flow along the river. But if you think of biological system, most of the time those systems are active systems, meaning that they are always out of equilibrium, and you have and the system is using the energy of the environment to perform some very particular motion. And this is something that you can find at the molecular level, at the cellular level, but as well at larger level as well. So if you can see here, for instance, you have those molecules that can spontaneously move and form this kind of weird shape. But if you take cells now also, they can move, and spontaneously they can get this kind of particular rotation, which is really due to the fact that they interact with each other. And maybe one of the good examples that clearly characterize our research is the intestinal organization. So if you look at the intestine, for instance, you have cells that are created, proliferate with the stem cell niche here. They proliferate along the villi here and they migrate. And on the top of the villi, where you have a high curvature, you have a huge extrusion of multiple cells. And interestingly, if you think of this mammalian intestine, it's covered by this single layer, as I was mentioning, and the renewal of cells is every 4 or 5 days. So this mechanism, the migration, but also the extrusion of cells is very important in vivo. And so this is typically 2 examples that I would like to give you. What we are doing is this idea of collective cell migration, but also the fact that cells can be extruded and how mechanics can play a role in this extrusion. Of course, both mechanisms are very important in cancer-related research, because if they are misregulated, it can lead to tissue invasion. So that's, for instance, one example. We are trying to engineer particular geometries, as you can see here, these kind of rings here. And what we observe is that spontaneously, again, when you plate cells on those structures, on this geometrical structure, then they start to rotate. So we understand how they move together, why they are rotating, and so on and so forth, and try to also link the molecular aspect to this multicellular dynamics. Another example that I would like to show is that we like to make analogies between biological system and physics. And for instance, here you have like cells that can actually interact with each other, and at the bottom part you have liquid crystal. And we managed to, by making analogies, so of course it's like some kind of sometimes starting point of the way of thinking research. By making analogy between cellular system and liquid crystal, we were able to show that the fingerprint of cell extrusion was really linked to the emergence of this particular structure, which are singularities, which are very well known in liquid crystal and which are called topological defects. And that those topological defects, in particular those structures here, they can lead to high forces, high compressive forces that could lead to extrusion of cells, as I can show you on this example here, where you can see you have flows around this tissue, and one cell can be extruded. The bright cells here is extruded, and is extruded exactly at a place where you found this topological defect. So of course, again, this research would have not been done by the help of many people. So this is still the group in Paris, and that on their move to, I mean, not all of them, but on their move to Erlangen. Thank you for your attention.
Yeah, so Benoit started by saying that he's French, so I have to reveal that I was born and raised in Iran. And Jochen also told you that this whole thing has cost about 60 million to the Bavarian government. This morning, my wife told me that I'm a very good proof for AfD's complaint that foreigners cost the government a lot of money. I'm very sorry about that. All right, the work that connects my research to the center is in the area of nanoBiophotonics. My real kind of origin is in atomic physics and quantum optics, and through the past 20, 30 years, I've drifted to develop a certain, let's say, expertise in optical microscopy and nanoscience, and through that we've realized that we can possibly contribute to biophysics and biomedical research. So there's this bridge, and right now the group actually has really 2 legs. One is on molecular quantum optics, and the other one, which is newer and is in nanobiophysics. So we are more methods developers. Jochen promoted developing methods by a quote from Sidney Brenner. So we've been developing various methods over the last 2 decades. On the left, you see detection of single proteins without needing to label them with fluorescence. This is an extremely sensitive way of detecting proteins using interferometry and looking at the scattering from proteins. In the second column, you see something completely different. This is cryogenic light microscopy, somewhat in parallel to cryogenic electron microscopy. You will have later a talk by Stefan Hell, who's the champion of optical resolution, but I can tell you that the resolution we have here is 10 times better than what they can do. So we're reaching really 3D angstrom resolution in a cryostat. Then we have developed a method for sizing and characterizing nanoparticles. We call it INTA. We're quite excited about that. For example, looking at exosomes and extracellular vesicles that Randy talked about. And more recently, we've developed a method that we call microkiss, as you see how the cell is being kissed, and I'll say something about that in a second. So the program that I want to introduce to you today as kind of one of the main directions in my group right now is viral infection. And so viral infection, usually one thinks has about 3 important phases. One of them is entry. Now with this movie that I stole from YouTube, you can see kind of how complicated that could be. There are lots of biochemical type of signaling and also physical processes that are involved when the virus first sees the cell membrane, and at some point it has to go in. So there's a lot of information there, and most of that is still unknown. I mean, you might have a vaccine, but we still don't know exactly how each of those processes in the cartoon really work. And this is a little bit of a problem with biology. There are lots of cartoons that make you think they know what they're doing, but actually most of the cartoons are really cartoons. Once the virus gets inside, then there's a process of reproduction. There's a lot of genetics. There's a lot of biochemistry again, and this is not something that we concentrate on as much. But when the virus wants to, when the cell has produced lots of viruses, these are ejected somehow out of the cell, and the process of exit is again a very interesting process. It happens somehow, it has to come out of the membrane, and then of course you have a whole bunch of them, and then this cell infects the neighboring cells. And we've developed, we really want to visualize and characterize every single step at the nanometer scale. That's kind of the passion that we have right now. I'd say probably will keep us busy for the next 10 years. And we have developed different bits and pieces of methods to tackle that. And one of them is this microkiss. So this is a method that we've developed by which we can really deliver individual particles like viruses or extracellular vesicles to a cell under microscope, even if it has to be a biosafety 2 lab. And in this movie, you can see how when I run it right at the edge of the yellow arrows, you will see pink spots. These pink spots are individual viruses that we, on command, introduce to the cell, and now we know exactly when these viruses are introduced, so we can chase them and see what happens to the life of the cell. We can look at the motion of these viruses on the membrane with incredible spatial and temporal resolution. What you see here is, for example, a trajectory of virus. You look at the length scale, you see that that length scale is 100 nanometers, so we really can watch the virus move with nanometer steps and really fast, of the order of hundredth of a millisecond. And that will help us understand how the virus actually interacts with the cell. And then once I go now to the exit, this is an example of what we have already done, where you see a cell. This is now fluorescence-free, kind of label-free microscopy. The dark spots that you see are viruses. You see that there are a lot of viruses, so this is when the cell has reproduced after infection. And we can watch how this cell is actually pushing these viruses out. This is the basal side of the cell, so we can look at this. These are just a very preliminary type of things that we've done, and now the program is to really try to understand the full virus life cycle. These are the people who are working currently in the group in the nanobiophotonics section, and I'd like to thank them very much for their hard work and you for listening.
It's my great pleasure to introduce our group who work on theoretical immunophysics. And by the background, we are theorists, but we belong officially to the Department of Biology at FAU. And what we do, we develop theoretical models which really help to understand complex biological phenomena and then their implications in the disease. And one can ask what type of the phenomena these are, and our spectrum is rather broad. I give only a few examples, like starting from describing the dynamics of immune cells which guard our tissues from pathogens, or looking how individual cells move and form cellular aggregates, or in a more kind of theory-oriented approaches to understanding the dynamics of general active biological systems, or looking at the phenotypic changes in multicellular bacterial aggregates, which are known as biofilms and considered to be models of tissues. So we have heard in principle that physics really is indispensable for the complete understanding for many complex biological processes, but also of the disease. But there's one very valid question, how actually theory can help. And to answer this question, I'm going to give one example. That's an example of the process of phase separation. So now we are going from supermarket home cooking. And what you're seeing here is this very famous demixing of oil and vinegar. So when you try to make a salad dressing, the stubborn vinegar droplets do not want to mix, so they phase separate. And this concept of phase separation essentially now is a very prominent concept in cell biology, which describes molecular organization of many things happening inside every cell of our body, but not only things inside of the cells go according to the phase separations, but also some cells do phase separate. And what we see in this movie, that's the clustering of individual necessary gonorrhea bacteria into huge aggregates containing up to several thousands of cells. And when this aggregate forms actually on the layer of epithelia in particular places in human, then this is when the symptoms of the second most common sexually transmitted disease, gonorrhea, start to develop. And we, of course, want to, on the one hand, model this process, and we can do that, but given the connection to the phase separation, which has really significant theory behind it, we can really put forward the equations which are going to describe this process of aggregation of the bacteria. So which means that with this, we have a really ground understanding of all the physical forces which drive the formation of such aggregates. And now that we understand that, we can cast our net a little broader and look at other systems which might behave similarly. In another movie, which I'm going to show you, is another process of aggregation. You see blue cells aggregating with the yellow ones, and the blue ones are actually the T cells, and the yellow ones are the dendritic cells. And what is happening on this movie is one of the very fundamental processes in immunology, where the dendritic cells, which have found some antigen of a pathogen, run to the T cells and say, look, I found something really bad. They show it to the T cells. T cells get activated, and then they go and kill essentially the cells which are exhibiting this type of the antigens on their surface. And that was happening here, so biologically very different from the aggregation of the bacterial cells. But if you look at the clustering dynamics, it was essentially very, very similar. So we now know which type of the forces should lead to this type of an aggregation, and from that we can actually go ahead and learn much better how an individual dendritic cell actually interacts with a cloud of T cells to activate them and do their job. So in general, therefore, we can say what theoretical models can help to do. They can really help us to see analogies between very, very biologically distinct systems. So they also, at the same time, because they're based on physics, which can be cast in the equations, allow us to provide very robust predictions about what kind of behaviors at all these biological systems can exhibit. And we're going to continue this type of a program. So probably I became too much of a biologist. There's going to be only cartoons, Vahid, of what we're going to do. So we're going to look at the pathogens, but in particular, we want to know how really physics affects the life of pathogens, and moreover, we want to interface those with the immune system of the host, and again focus on how physical interactions affect the process of infection, the process of inflammation, and then eventually the resolution of the inflammation. And therefore, really coupling physics and immunology, we really push this field of immunophysics. So with that, thanks for your attention.